Chapter 1. Early Life. Carl Woese was born on July 15, 1928, in Syracuse, New York. Syracuse is a city known for its cold winters and warm summers. Carl grew up in a modest house on a quiet street. His father, Edward, worked as an engineer, and his mother, Gertrude, was a teacher. Carl had a younger sister named Alice. They were a close-knit family, spending many evenings together, sharing stories and playing games. From a young age, Carl was curious about the world around him. He was always asking questions. Why is the sky blue? How do birds fly? His parents encouraged his curiosity. They believed in the importance of education and always tried to answer his questions, or they would find books that could. When Carl was 5 years old, he received a small microscope for his birthday. It was a simple toy, but for Carl, it was a gateway to a new world. He spent hours looking at tiny insects, leaves, and even drops of water. Every time he looked through the microscope, he discovered something new. It was as if there was a whole universe hidden in the small things around him. Carl started school when he was 6. He was a bright student and loved learning. His favorite subjects were science and math. He enjoyed the logical thinking required in math and the mysteries he could solve with science. He liked to do experiments at home, sometimes making a mess in the kitchen, much to his mother's dismay. But she never scolded him too much because she saw how passionate he was. One winter, when Carl was 10, there was a big snowstorm. The schools were closed and the streets were covered in snow. Carl and his sister built a snowman and had snowball fights with the neighborhood kids. But after a few days, Carl grew restless. He missed school and his science books. One evening after dinner, Carl's father brought home an old radio. It was broken, but he thought it might be interesting for Carl to try to fix it. Carl's eyes lit up with excitement. He had never fixed a radio before, but he was eager to learn. He took the radio apart, piece by piece, studying each component carefully. He asked his father questions about how the radio worked and why it was not working. After many hours of trial and error, Carl managed to fix the radio. The moment he turned it on and heard music coming from it, he felt a sense of accomplishment he had never felt before. It was in that moment that Carl realized he loved not just learning about how things worked, but also solving problems and fixing things. As Carl grew older, his love for science grew stronger. In high school, he excelled in his science classes. His teachers noticed his talent and encouraged him to pursue a career in science. Carl was fascinated by the idea of discovering new things and making a difference in the world. One summer, Carl had the opportunity to attend a science camp. It was a 2-week camp where students could do experiments and learn from real scientists. Carl was thrilled. At the camp, he met other students who shared his passion for science. They talked about their favorite experiments and scientists they admired. For the first time, Carl felt like he was part of a community of people who loved science as much as he did. At the camp, Carl learned about different fields of science. He was particularly interested in biology. He was fascinated by the complexity of living organisms and how everything in nature was interconnected. One of the camp instructors, a biologist, showed Carl how to collect samples from a nearby lake and look at them under a microscope. Carl was amazed at the diversity of life he could see in just a drop of water. Back at school, Carl continued to excel. He joined the science club and participated in science fairs. He loved the challenge of coming up with new experiments and presenting his findings. His teachers and classmates were impressed by his knowledge and enthusiasm. When it was time to think about college, Carl knew he wanted to study science. He applied to Amherst College, a small liberal arts college in Massachusetts, known for its strong science program. He was accepted and received a scholarship to help with the costs. Leaving home for the first time was both exciting and scary for Carl. He would miss his family, but he was eager to start this new chapter in his life. At Amherst, Carl immersed himself in his studies. He took classes in biology, chemistry, and physics. He spent long hours in the library and the laboratory, always eager to learn more. College was a transformative experience for Carl. He met professors who were experts in their fields and who inspired him to think critically and ask big questions. He also met fellow students who were just as passionate about science as he was. They would often stay up late discussing their ideas and dreams for the future. One of Carl's professors, Dr. Johnson, noticed his exceptional talent and dedication. Dr. Johnson became a mentor to Carl, offering guidance and support. He encouraged Carl to pursue his own research projects and introduced him to new areas of study. Under Dr. Johnson's mentorship, Carl began to explore the field of molecular biology. He was fascinated by the idea that the secrets of life could be found at the molecular level. Carl's time at Amherst laid the foundation for his future career. He graduated with honors, ready to take on the next challenge. His curiosity and passion for science had only grown stronger, and he was eager to continue his journey of discovery. As Carl Woese moved on to the next stage of his life, he carried with him the lessons he had learned from his early years. His family's support, his early experiments, and his college experiences had all shaped him into a determined and curious scientist, ready to make his mark on the world. Chapter 2. College Years. Carl Woese arrived at Amherst College in the fall of 1945. The campus was beautiful, with its old brick buildings and large green lawns. Carl felt a mix of excitement and nervousness as he walked through the gates. This was the beginning of a new adventure, and he was eager to dive into his studies. Carl had always been passionate about science, and at Amherst, he decided to focus on biophysics. Biophysics is a field that combines biology and physics to understand how living things work. It was the perfect subject for Carl, who loved both the living world and the laws that governed it. His first year at college was challenging. The classes were harder than anything he had experienced in high school. He had to study long hours to keep up with the coursework. His days were filled with lectures, lab sessions, and hours spent in the library. But Carl didn't mind the hard work. He enjoyed the challenge and was determined to succeed. One of his favorite classes was Introduction to Molecular Biology. The professor, Dr. Simmons, was a well-respected scientist with a deep knowledge of the subject. He made the complex topics understandable and exciting. Carl was fascinated by how tiny molecules inside cells could control so many processes in living organisms. In the evenings, Carl often studied with his friends. They would gather in the common room of their dormitory, books and notes spread out on the tables. They quizzed each other, discussed difficult concepts, and shared their dreams for the future. It was during these study sessions that Carl realized he wasn't alone in his passion for science. He had found a community of like-minded people who inspired and motivated him. Despite his busy schedule, Carl made time for other activities. He joined the college's science club, where he met students from different disciplines. They organized talks, invited guest speakers, and even went on field trips to local laboratories. These activities broadened Carl's perspective and introduced him to new areas of science. One memorable field trip was to a nearby research lab that specialized in microbiology. Carl had always been curious about the microscopic world, and this visit sparked a new interest. He saw researchers working with powerful microscopes, examining bacteria and viruses. The idea that such tiny organisms could have such a big impact on the world fascinated him. Back at Amherst, Carl decided to learn more about microbiology. He started reading books and scientific papers on the subject. He was amazed at how much there was to learn and how many mysteries were still unsolved. His curiosity grew, and he began to think about how he could contribute to this field. In his second year, Carl faced a major challenge. He had to take a difficult physics course that many students struggled with. The professor, Dr. Hayes, was known for his tough exams and high expectations. Carl knew he would have to work harder than ever to do well in this class. He spent countless hours in the physics lab, conducting experiments and analyzing data. There were times when he felt frustrated and doubted his abilities. But he didn't give up. He asked for help when he needed it, and he studied with his friends, who were also taking the class. Together, they tackled the difficult problems and supported each other. One day, Carl was working on a particularly challenging experiment. He had tried several times to get the results he needed, but something kept going wrong. He felt like giving up, but then he remembered his father's words. Problems are opportunities in disguise. With renewed determination, Carl went back to the drawing board, rechecking his calculations and carefully adjusting his equipment. After several more attempts, he finally succeeded. The data was perfect, and Carl felt a surge of triumph. This experience taught him an important lesson about perseverance and problem solving. It was a moment that would stay with him throughout his career. As Carl progressed through college, his hard work began to pay off. He received high grades and earned the respect of his professors. Dr. Simmons, in particular, took notice of Carl's dedication and talent. He invited Carl to join his research team, giving him the opportunity to work on real scientific projects. Working in Dr. Simmons' lab was an eye-opening experience. Carl learned how to design experiments, analyze results, and write scientific papers. He was involved in a project studying the structure of DNA, the molecule that carries genetic information in living organisms. This was cutting-edge research, and Carl was thrilled to be a part of it. In his third year, Carl had to choose a topic for his senior thesis. This was a major project that would take up much of his time and energy. After much thought, he decided to focus on the role of ribosomes in protein synthesis. Ribosomes are tiny structures in cells that make proteins, which are essential for life. Carl spent months researching and conducting experiments. He read countless scientific papers and spent long hours in the lab. There were times when he felt overwhelmed by the amount of work, but he was driven by his passion for discovery. He wanted to contribute something meaningful to the field of biophysics. His hard work paid off. At the end of his senior year, Carl presented his thesis to a panel of professors. They were impressed by his research and the depth of his understanding. Carl felt a sense of accomplishment and pride. He had overcome many challenges and achieved something significant. Graduation day was a memorable event. Carl's family traveled to Amherst to celebrate with him. As he walked across the stage to receive his diploma, he thought about all the hard work and determination it had taken to get to this point. He felt grateful for the support of his family, friends, and professors who had helped him along the way. Carl Woese left Amherst College with a solid foundation in biophysics and a growing interest in microbiology. His college years had been a time of intense learning and personal growth. He had faced challenges, made discoveries, and forged lasting friendships. Most importantly, he had found his calling in the world of science. As he looked ahead to the future, Carl felt a sense of excitement and anticipation. He knew that there were many more discoveries to be made and challenges to be faced. With his passion for science and the skills he had gained at Amherst, he was ready to take the next step in his journey. Chapter 3: Early Career. Carl Woese graduated from Amherst College with honors and a head full of dreams. It was the early 1950s, a time when science was making great strides. The excitement in the air was palpable, and Carl was eager to begin his career. His first step after college was to continue his studies. Carl was accepted into the PhD program at Yale University, one of the most prestigious universities in the United States. At Yale, Carl focused on biophysics, a field that combined his love for both biology and physics. The campus was buzzing with activity, and Carl was thrilled to be part of it. At Yale, Carl's days were filled with lectures, research, and experiments. He worked in a lab with other students, each of them driven by a desire to uncover the mysteries of life. Carl's research focused on the structure of proteins. Proteins are essential molecules in all living organisms, and understanding them was key to many scientific breakthroughs. One of Carl's early challenges was learning how to use new and complex equipment. The lab had state-of-the-art machines that could analyze proteins at a molecular level. Carl spent many hours learning how to operate these machines, often staying late into the night. His dedication was evident, and his professors took notice. During his time at Yale, Carl made significant progress in his research. He discovered new methods for analyzing proteins, which would later prove to be valuable in his future work. His hard work and innovative thinking earned him respect among his peers and professors. After completing his PhD, Carl faced a critical decision: where to go next. He received an offer to work at General Electric, GE Research Laboratory. GE was not just an industrial giant, it was also a hub for scientific research and innovation. Carl saw this as a unique opportunity to apply his knowledge in a practical setting. At GE, Carl joined a team of scientists working on various projects. His role was to study the molecular structure of different materials. This work was different from his previous research, but Carl was excited about the new challenge. He believed that understanding the molecular structure could lead to significant advancements in technology and industry. One of Carl's first projects at GE was to study polymers. Polymers are long chains of molecules that make up many materials, such as plastics. Understanding their structure was crucial for developing new and better materials. Carl used his skills in molecular biology to analyze these polymers and provide insights that helped improve their quality and functionality. Carl's work at GE was both demanding and rewarding. He had to learn new techniques and adapt to a different environment. He worked closely with engineers and other scientists, which broadened his perspective and enhanced his problem-solving skills. The experience taught him how to apply scientific principles to real-world problems, a lesson that would be valuable throughout his career. Despite the challenges, Carl found time to pursue his own research interests. He continued to study proteins and their role in living organisms. He was particularly interested in how proteins are made in cells. This process, known as protein synthesis, was a mystery that many scientists were trying to unravel. In his spare time, Carl read extensively about molecular biology. He was fascinated by the idea that tiny molecules could control so many aspects of life. This fascination fueled his curiosity and drove him to explore new research areas. During this period, Carl developed a strong network of colleagues and mentors. He attended scientific conferences and seminars where he met other researchers who shared his interests. These interactions sparked new ideas and collaborations, further enriching his research. One pivotal moment in Carl's early career came when he attended a lecture by Dr. James Watson, one of the discoverers of the structure of DNA. DNA, or deoxyribonucleic acid, is the molecule that carries genetic information in living organisms. Dr. Watson's lecture opened Carl's eyes to the possibilities of molecular biology. He realized that understanding DNA could unlock the secrets of life itself. Inspired by this lecture, Carl decided to focus his research on genetic information and its role in protein synthesis. He began to investigate how DNA directs the production of proteins in cells. This line of research was still in its early stages, and Carl saw an opportunity to make significant contributions. At GE, Carl conducted experiments to study how DNA and proteins interact. He used advanced techniques to analyze these interactions and gather data. His findings provided new insights into the molecular mechanisms of life. Carl's work was recognized by his peers, and he published several influential papers on the subject. Carl's time at GE was a period of intense learning and growth. He gained valuable experience in both basic and applied research. He learned how to communicate his findings effectively, both in writing and in presentations. These skills would serve him well in his future career. As Carl's reputation grew, he received offers from various universities and research institutions. He decided to accept a position at the University of Illinois at Urbana-Champaign. This move marked a new chapter in his career, one that would lead to groundbreaking discoveries. At the University of Illinois, Carl continued his research on molecular biology. He was excited about the opportunities to teach and mentor young scientists. He believed in the importance of education and was eager to inspire the next generation of researchers. Carl's early career was marked by determination, curiosity, and a relentless pursuit of knowledge. He faced many challenges, but always found ways to overcome them. His work at Yale and GE laid the foundation for his future achievements. Carl Woese was on the path to becoming a pioneer in molecular biology, ready to make discoveries that would change our understanding of life. As he looked ahead, Carl knew that the journey would not be easy. But with his passion for science and his unwavering dedication, he was prepared to face whatever challenges came his way. His early career had taught him that with hard work and perseverance, anything was possible. And so, Carl Woese embarked on the next phase of his journey, ready to unlock the secrets of the molecular world. Chapter 3. Early Career. Carl Woese graduated from Amherst College with honors and a sense of excitement about his future. It was the early 1950s, a time when science was rapidly advancing, and Carl was eager to contribute to these discoveries. His journey led him first to Yale University for his PhD studies. At Yale, Carl dove deep into the field of biophysics, which combines biology and physics to understand how living things work. Yale was a prestigious university, and Carl felt a mix of excitement and nervousness as he joined the campus. He knew that he had to work hard to succeed in this challenging environment. Carl's days at Yale were filled with lectures, lab experiments, and long hours of studying. One of his favorite courses was molecular biology, taught by Dr. Simmons, a renowned scientist. Dr. Simmons made complex topics understandable and exciting, and Carl was fascinated by how tiny molecules inside cells could control so many processes in living organisms. In the lab, Carl worked with other students on various experiments. They were studying proteins, which are essential molecules in all living organisms. Carl was particularly interested in understanding how proteins are made inside cells. This process, called protein synthesis, was a mystery that many scientists were trying to solve. During his time at Yale, Carl faced many challenges. Learning to use new and complex equipment was difficult. He spent countless hours in the lab, often staying late into the night to perfect his experiments. His dedication and hard work paid off as he made significant progress in his research. Carl discovered new methods for analyzing proteins, which would later prove valuable in his career. After earning his PhD, Carl faced an important decision: where to work next. He received an offer from General Electric, GE Research Laboratory. GE was a major company known for its innovation and scientific research. Carl saw this as an opportunity to apply his knowledge in a practical setting. At GE, Carl joined a team of scientists working on various projects. His role was to study the molecular structure of different materials. This work was different from his previous research, but Carl was excited about the new challenge. He believed that understanding molecular structures could lead to significant advancements in technology and industry. One of Carl's first projects at GE was to study polymers. Polymers are long chains of molecules that make up many materials, such as plastics. Understanding their structure was crucial for developing new and better materials. Carl used his skills in molecular biology to analyze these polymers and provide insights that helped improve their quality and functionality. Working at GE was demanding but rewarding. Carl had to learn new techniques and adapt to a different environment. He worked closely with engineers and other scientists, which broadened his perspective and enhanced his problem-solving skills. The experience taught him how to apply scientific principles to real-world problems, a lesson that would be valuable throughout his career. Despite the challenges, Carl found time to pursue his own research interests. He continued to study proteins and their role in living organisms. He was particularly interested in how proteins are made in cells. This process, known as protein synthesis, was a mystery that many scientists were trying to unravel. In his spare time, Carl read extensively about molecular biology. He was fascinated by the idea that tiny molecules could control so many aspects of life. This fascination fueled his curiosity and drove him to explore new research areas. During this period, Carl developed a strong network of colleagues and mentors. He attended scientific conferences and seminars where he met other researchers who shared his interests. These interactions sparked new ideas and collaborations, further enriching his research. One pivotal moment in Carl's early career came when he attended a lecture by Dr. James Watson, one of the discoverers of the structure of DNA. DNA, or deoxyribonucleic acid, is the molecule that carries genetic information in living organisms. Dr. Watson's lecture opened Carl's eyes to the possibilities of molecular biology. He realized that understanding DNA could unlock the secrets of life itself. Inspired by this lecture, Carl decided to focus his research on genetic information and its role in protein synthesis. He began to investigate how DNA directs the production of proteins in cells. This line of research was still in its early stages, and Carl saw an opportunity to make significant contributions. At GE, Carl conducted experiments to study how DNA and proteins interact. He used advanced techniques to analyze these interactions and gather data. His findings provided new insights into the molecular mechanisms of life. Carl's work was recognized by his peers, and he published several influential papers on the subject. Carl's time at GE was a period of intense learning and growth. He gained valuable experience in both basic and applied research. He learned how to communicate his findings effectively, both in writing and in presentations. These skills would serve him well in his future career. As Carl's reputation grew, he received offers from various universities and research institutions. He decided to accept a position at the University of Illinois at Urbana-Champaign. This move marked a new chapter in his career, one that would lead to groundbreaking discoveries. At the University of Illinois, Carl continued his research on molecular biology. He was excited about the opportunities to teach and mentor young scientists. He believed in the importance of education and was eager to inspire the next generation of researchers. Carl's early career was marked by determination, curiosity, and a relentless pursuit of knowledge. He faced many challenges but always found ways to overcome them. His work at Yale and GE laid the foundation for his future achievements. Carl Woese was on the path to becoming a pioneer in molecular biology, ready to make discoveries that would change our understanding of life. As he looked ahead, Carl knew that the journey would not be easy. But with his passion for science and his unwavering dedication, he was prepared to face whatever challenges came his way. His early career had taught him that with hard work and perseverance, anything was possible. And so, Carl Woese embarked on the next phase of his journey, ready to unlock the secrets of the molecular world. Chapter 4. Discovering Ribosomal RNA. Carl Woese arrived at the University of Illinois at Urbana-Champaign with a sense of purpose. He was determined to continue his research in molecular biology, but he had no idea that he was about to make one of the most significant discoveries in the history of science. Carl had always been fascinated by the idea of understanding life at a molecular level. He believed that by studying the smallest parts of living organisms, scientists could uncover the secrets of life itself. At the University of Illinois, he had access to state-of-the-art laboratories and equipment, which fueled his curiosity and ambition. One day, while reading a scientific paper, Carl came across an intriguing idea. The paper suggested that ribosomal RNA, rRNA, a type of RNA found in all living cells, could be used to study the evolutionary relationships among organisms. This was a revolutionary concept. Scientists knew that rRNA played a crucial role in protein synthesis, but no one had considered using it to explore evolution. Carl was captivated by this idea. He thought that if he could analyze the rRNA sequences from different organisms, he might be able to trace their evolutionary history. This could provide a new way to understand how life on Earth had evolved. Determined to pursue this idea, Carl began to design experiments to study rRNA. He knew that this would be a challenging task. Analyzing rRNA sequences was a complex process that required advanced techniques and careful precision. But Carl was not discouraged. He was excited by the challenge and eager to see where this new line of research would lead. In his lab, Carl developed a method to isolate rRNA from different organisms. He collected samples from bacteria, plants, and animals, carefully extracting the rRNA from each one. This process was time-consuming and required meticulous attention to detail. Carl spent long hours in the lab, often working late into the night. Once he had isolated the rRNA, Carl began to analyze the sequences. He compared the rRNA sequences from different organisms, looking for similarities and differences. It was a painstaking process, but Carl's determination never wavered. He knew that if he could find patterns in the rRNA sequences, he might be able to uncover the evolutionary relationships among organisms. As Carl analyzed the rRNA sequences, he began to see patterns emerge. He noticed that certain parts of the rRNA sequence were highly conserved, meaning they were similar across different organisms. These conserved regions suggested that these organisms shared a common ancestor. Other parts of the rRNA sequence were more variable, indicating evolutionary changes that had occurred over time. Carl realized that these patterns could be used to construct a tree of life that showed the evolutionary relationships among organisms. This was a groundbreaking idea. By using rRNA sequences, Carl could trace the evolutionary history of life on Earth in a way that had never been done before. One of Carl's most significant findings was the discovery of a new group of organisms, which he called archaea. The rRNA sequences from these organisms were distinct from those of bacteria and eukaryotes, organisms with complex cells. This suggested that archaea represented a separate branch on the tree of life, distinct from the other two groups. This was a revolutionary discovery that challenged the traditional view of the classification of life. Carl's discovery of archaea had profound implications for the field of biology. It showed that life on Earth was more diverse than previously thought, and that the evolutionary history of organisms was more complex. This discovery opened up new avenues of research and led to a deeper understanding of the origins and evolution of life. Carl's work on rRNA was met with excitement and skepticism. Many scientists were intrigued by his findings, but others were skeptical of his methods and conclusions. Carl knew that he needed to provide robust evidence to support his claims. He continued to refine his techniques and gather more data, determined to convince the scientific community of the validity of his work. Over time, Carl's research gained widespread acceptance. His methods for analyzing rRNA sequences became standard practice in the field of molecular biology. Scientists around the world began to use rRNA to study the evolutionary relationships among organisms, and Carl's work became a cornerstone of modern biology. Carl's discovery of rRNA and its significance in studying evolution had a profound impact on the scientific community. It provided a new tool for exploring the origins and diversity of life, and revolutionized the way scientists understood evolutionary relationships. Carl's work opened up new possibilities for research and paved the way for future discoveries. As Carl looked back on his journey, he felt a deep sense of satisfaction. He had faced many challenges and obstacles, but his determination and passion for science had driven him to make one of the most significant discoveries of his career. Carl woese's work on rRNA had not only advanced the field of molecular biology, but had also deepened our understanding of the history of life on Earth. Carl's story is a testament to the power of curiosity and perseverance. His groundbreaking discovery of rRNA and its role in studying evolution has left a lasting legacy in the field of science. As future generations of scientists continue to explore the mysteries of life, they will build on the foundation laid by Carl Woese and his pioneering research. In the years that followed, Carl continued to explore new questions and challenges in molecular biology. His work inspired countless scientists and students, encouraging them to pursue their own discoveries. Carl woese's legacy lives on in the ongoing quest to understand the fundamental principles of life and the intricate web of relationships that connect all living things. Chapter 5. The Three-Domain System. Carl woese's groundbreaking discovery of ribosomal RNA, rRNA, had opened new doors in the study of life's evolution. But Carl was not one to rest on his laurels. He was constantly driven by curiosity and a desire to understand the fundamental principles of life. His next big idea would revolutionize the way scientists viewed the tree of life. In the late 1970s, Carl was deeply immersed in his research at the University of Illinois. He had spent years analyzing rRNA sequences from various organisms, and the patterns he observed were intriguing. He noticed that some rRNA sequences were unlike anything seen before. These sequences did not fit neatly into the known categories of life: prokaryotes, which include bacteria, and eukaryotes, organisms with complex cells like plants and animals. Carl began to suspect that there was a third, distinct group of organisms. This idea was revolutionary and controversial. Up until then, scientists had classified all life into two main groups: prokaryotes and eukaryotes. Carl's research suggested that this classification was incomplete. To explore this idea further, Carl and his team continued to collect and analyze rRNA sequences from a wide range of organisms. They used advanced techniques to isolate and sequence the rRNA, meticulously comparing the results. The more data they gathered, the clearer the picture became. There was indeed a third group of organisms that were fundamentally different from both bacteria and eukaryotes. Carl named this third group Archaea. The Archaea had unique rRNA sequences that set them apart from bacteria. This was a groundbreaking discovery. It meant that the tree of life had three main branches, not just two. Carl proposed a new classification system, which he called the three-domain system. According to this system, all life on Earth could be classified into three domains: bacteria, archaea, and eukarya. The three-domain system was a bold and innovative idea. It challenged the traditional view of the tree of life and introduced a new way of thinking about evolutionary relationships. Carl's proposal was met with a mixture of excitement and skepticism. Some scientists were intrigued by the idea, while others were doubtful. They questioned the methods and the conclusions, unsure whether to accept this radical new system. Carl knew that convincing the scientific community would not be easy. He published his findings in scientific journals, detailing the evidence that supported his three-domain system. He presented his research at conferences, explaining how the unique rRNA sequences of archaea justified their classification as a separate domain. He engaged in debates and discussions, addressing the concerns and criticisms of his peers. Despite the initial resistance, Carl's persistence began to pay off. More and more scientists started to recognize the validity of his work. Researchers began to find additional evidence supporting the existence of the archaea. They discovered that these organisms thrived in extreme environments, such as hot springs and salt lakes, further distinguishing them from bacteria and eukaryotes. The acceptance of the three-domain system had profound implications for the field of biology. It provided a new framework for understanding the diversity of life on Earth. Scientists could now study evolutionary relationships with a clearer perspective, identifying the unique characteristics of each domain. The discovery of archaea also sparked new interest in exploring extreme environments, leading to further discoveries about the resilience and adaptability of life. Carl's work on the three-domain system extended beyond the scientific community. It captured the imagination of the public, inspiring curiosity about the origins and diversity of life. People began to see life on Earth as a more complex and interconnected web, with three main branches that had evolved over billions of years. One of the most significant impacts of Carl's work was on the study of evolution. The three-domain system provided new insights into how different forms of life had evolved from common ancestors. It helped scientists trace the evolutionary history of organisms with greater accuracy, revealing the intricate relationships between species. This new understanding of evolution had far-reaching implications for various fields, including medicine, ecology, and biotechnology. Carl's discovery also highlighted the importance of molecular biology in studying life's diversity. By analyzing molecules like rRNA, scientists could uncover hidden patterns and relationships that were not visible through traditional methods. Carl's work demonstrated the power of molecular techniques in unraveling the mysteries of life, paving the way for future research and discoveries. As the three-domain system gained acceptance, Carl woese's reputation as a pioneer in molecular biology grew. He received numerous awards and honors for his contributions to science. His work inspired a new generation of scientists, encouraging them to explore the frontiers of molecular biology and evolutionary research. Carl's journey was a testament to the power of curiosity and perseverance. He faced skepticism and criticism, but his dedication to uncovering the truth drove him forward. His revolutionary idea changed the way we understand the tree of life, providing a new framework for studying the diversity of life on Earth. The three-domain system remains a cornerstone of modern biology. It continues to guide research and discovery, shaping our understanding of life's complexity. Carl woese's legacy lives on in the work of scientists around the world who build on his discoveries to explore new questions and challenges. Carl's story is an inspiring example of how one person's curiosity and determination can change the course of science. His groundbreaking discovery of the three-domain system has left a lasting impact on the field of biology, reminding us of the endless possibilities that come from exploring the unknown. Chapter 6. Challenges and Controversies. Carl woese's proposal of the three-domain system was revolutionary, but not everyone in the scientific community was ready to accept it. The idea of classifying life into bacteria, archaea, and eukarya was new and challenging. Many scientists had spent their careers working with the older system, which divided life into just two groups, prokaryotes and eukaryotes. Carl's discovery meant rethinking much of what they thought they knew. At first, Carl was excited about his findings. He believed that the scientific community would see the evidence and understand the importance of his work. But soon, he realized that his new classification system was met with skepticism and resistance. Many scientists questioned his methods and doubted his conclusions. Some thought his ideas were too radical and did not fit with established knowledge. One of the biggest challenges Carl faced was convincing others that archaea were a distinct domain of life. Many scientists were not ready to accept that these microorganisms were fundamentally different from bacteria. They argued that the differences Carl had found in ribosomal RNA, rRNA sequences, were not enough to justify a new classification. Carl was determined to prove his findings. He knew that science often advanced through debate and questioning, and he was ready to defend his work. He continued to gather more data, analyzing rRNA sequences from a wider variety of organisms. The more he studied, the more evidence he found to support his three-domain system. Despite his growing body of evidence, the debates and controversies continued. Carl presented his research at scientific conferences, where he faced tough questions and criticisms. Some scientists challenged his methods, while others argued that his conclusions were premature. These meetings were often intense, but Carl remained calm and focused. He believed in his work and knew that science required patience and persistence. One memorable conference took place in the late 1970s. Carl presented his latest findings, showing detailed comparisons of rRNA sequences from different organisms. He explained how these sequences revealed deep evolutionary relationships that could not be seen through traditional methods. The room was filled with scientists, many of whom were skeptical. As Carl spoke, he could see some of them shaking their heads, while others listened intently. During the question and answer session, one scientist stood up and challenged Carl's conclusions. He argued that the differences in rRNA sequences were not significant enough to justify a new domain. Carl listened carefully and then responded with clear, detailed explanations. He pointed out the unique characteristics of archaea and how they differed from both bacteria and eukaryotes. By the end of the session, some in the audience were convinced, but many still had doubts. These debates were exhausting, but they also pushed Carl to refine his research and strengthen his arguments. He knew that convincing the scientific community would take time, and he was willing to put in the effort. He continued to publish his findings in scientific journals, providing more and more evidence to support his theories. Outside the scientific community, Carl faced additional challenges. Funding for his research was not always easy to secure. Many funding agencies were hesitant to support such a controversial idea.